I'll be speaking on our recent work in single-cell and spatial methods for understanding complex diseases. So I got my training in population genetics, and I just want to spend a few slides on some of the background information. So we know that GWAS has identified 24 top association in 2007, but now in 2026, there's more than 1 million top associations across 140 full summary statistics across more than 7,000 publications. So then our strategic priority has changed. In 2007, our goal was to identify more GWAS loci, but now our priority has been that there's too much association and we need to identify the target genes. But unfortunately, we're not doing very well on that, even after we have spent so much money on GTACs, which is a $100 million project, spanning almost a decade. We sequenced 50 tissues across almost 1,000 individuals, and we identified that almost every SNP in the human genome is associated with expression of at least one gene. But still, only less than 50% of GWAS loci can be explained by colocalizing with at least one GTAC tissue. So, and to give you a more concrete example, right? I take the example from cardiovascular diseases. So there's a number of consortiums working on this area. There's more than 600 CVD loci. This is statistics a few years back, so there's more now. But only 4.5% have validated target genes, and all of them I've listed on the right side. So how do we push this number from 4.5% to be a little bit higher, right? That's the main goal of my group. And we don't stand alone in this fight. There has been a number of other groups that's done fantastic studies alongside with us. So what people have converged on is that we not only need to see the tissues, we need to see the cell type specific regulation at the right differentiation stages and also at the right perturbations. And for us, our group is also interested in two additional things. One is the right ancestry and also the right molecular phenotypes. So in the past few years, efforts has been made in this area from people in Australia like Joseph Powell, Daniel MacArthur, and people in Europe, Lud Frank, and people in the US, and also locally in Singapore, to publish these papers on doing single-cell eQTL calling. Although these papers are very nice, they suffer from two same problems, right? They're only one population, which is the European population, and they only care about one phenotype, which is expression, right? So to address these two problems, we have started in collaboration with our very generous collaborators, Sham and Ken Hong. We have done single-cell RNA-seq of 1 million PBMCs from 500 donors of diverse Asian descent, right? So Singapore is it's a very small island, but it covers 96% of Asian genetic diversity, which is quite unique. So we have individuals from East Asian, Southeast Asian, and South Asians, and we sequenced more than 1 million cells across the 22 cell types. Some of you might recognize these cell types. So there's PDCs, there's dendritic cells, monocytes, natural killer cells, various types of T cells, and various types of B cells. So the first question that we asked, because back when we started this project in 2022, the common belief in the field is that single-cell sequencing, because it's single-end biased, it's either 5' biased or 3' biased, it's not really possible to do splicing based on single-cell data. And that's actually, that's still the prevailing belief today, because every time I gave this talk, they would say, are you using long read? In fact, we're not using long read. It's too expensive for the scale that we're doing. So we're using just regular short read single-cell sequencing data. So how are we able to get splicing information out of end biased single-cell splicing data? And that's the key story I'm going to tell you today. So previously, using the 1K study, they were able to get 267 alternatively spliced genes per cell. This is dashed line here. I should explain this figure. So x-axis is the different cell types, dendritic cells, monocytes, B cells, T cells, and natural killer cells. Y-axis is the number of alternatively spliced genes we detect per cell. Okay, so previously the state of the art is 267. So we were able to push this number up about 5-fold to be somewhere between 1,000 to 2,000. This is our data. So some of you might be asking, I'll explain how we do this later, but some of you might be asking, are we just sequencing more deeply? If we're sequencing more deeply, of course, we'll be detecting more genes, and therefore we'll be detecting more alternatively spliced genes. And that's true to some extent, but that's not the full story. So if you look at the figure on the right side, on the x-axis, that's the number of expressed genes. On the y-axis, that's the number of alternatively spliced genes. You see that there is a linear correlation between these two axes, right? But if you see the slope of the axis, our data in red is about 5 times higher, steeper than the 1K data in blue, which basically means that for the same read depth, we'll get more coverage of exons and more detection of alternatively spliced genes per cell. So how are we able to do this? So there's two ways, right? There's two complementary approaches that we did. The first one is that we use a what we call a 5' capture assay instead of the more commonly used 3' capture assay. So why does this make a difference? When you do 3' sequencing, you're capturing at the poly A site. So then basically every read 1 will start from poly A. So basically that means you start at a fixed 3' end. But if you do 5', there's two competing forces that cuts the 5' short. The force number 1 is infidelity of the PCR of the polymerase, right? So they would drop off at a random point at the 5'. This could be a bad thing, but in this case, it's a good thing because you start a random 5' site. And also intrinsically, there's a mechanism in every single human cell called stochastic cleavage and recapping. So basically, your cell will randomly cleave off the 5' end. This is a mechanism in your cell to increase cellular diversity for evolutionary purposes. It does not change the coding sequence, but it changes the 5' end, 5' UTR regions. So because of these two competing forces, you get a random 5' start. And then because we do fragmentation, you get a random 3' end. So now you're basically doing RNA-seq, right? Even though it's 5' biased, but it's more random. So in the end, what we found is that more than 80 single-cell RNA-seq data using 5' sequencing method can cover more than 80% of transcripts on average, of exons on average. Okay. And also, I'm not showing here is that we validated using. Actually, let me just show you the data. I think I hide the slide. Okay. So then are we confident in our splice junction calling? And the answer is yes, because we validated this using long read data, right? And just using 4 individuals compared to the 500 individuals we did short read, we were able to recover more than 97% of all the splice junctions that we recover using short read. Okay. The novel splice sites tend to be cell type specific splice sites, which basically means that they're not they could be false positives, but what we see is that the novel splice sites that are not covered by the long reads, they're incredibly cell type specific, so they're just not captured in our long reads data. Okay, so once we have this splice junction reading, what we can do is that we can compare across populations, right? So here I would like to pose a question for you guys. Usually when we do differential splicing, we usually would compare a healthy population against a disease population. But here we're actually comparing healthy population against other healthy populations. And we still found great many differential splicing events. So right? you're, two healthy person, should have relatively the same gene expression and relatively the same splicing transcripts, but still we found a great number of differential splicing events. So what we found is that these differential splicing events is actually driven by the allele frequency differences. So I'll give you one example. In this gene SPSP2, we found that there is a genetic variance right here, RS110, that changes the AGGA splice site to an AGAA, deactivating the splice site, which forces the spliceosome to choose to scan the transcript and find an alternative splice site 12 BP upstream, which is AGGA. So that's a cryptic splice that's not annotated, right? So this is the new splice isoform, the red one, and it's actually not showing up in gene code. Okay, so what's more interesting is that this variance, this minor allele frequency, is graded across the three populations. It's the highest in East Asians, medium in Southeast Asians, and lowest in South Asians. So then what we see is that the splicing pattern corresponded beautifully with this minor allele frequency gradient. So it's the canonical splice site is used least frequently in the East Asians because the splice site is destroyed in most of the East Asian people. Medium in Southeast Asian people, and the highest in South Asian people. So because we had genetic data, we can also do splicing QTL calling. So we identified more than 11,000 cis sQTLs and also 607 trans QTLs. So I'll give you two examples using our splicing QTL data. The first example we have is on the biological pathways. So we're able to identify two distinct variants in two different cell types regulating the same gene. So to be more specific, we identified these two variants, this red one and this purple one, in naive CD4 T cells and memory CD4 T cells, regulating the same gene. Okay, we know that these two variants are independent with a high degree of confidence because of two reasons. The first one is that these two variants, RS675 and RS742, they're 36 kb apart, right? So much longer than an enhancer would cover. And also their R squared is quite low. Their R squared is 0.28, okay? So they're very likely to be independent. That's basically saying that, even if there's genetic regulatory mechanisms in two cell types, sometimes it's different cis regulatory elements that's regulating the same gene expression. What's more interesting is that these two exact same lead variants are also splicing QTLs in trans or different gene. So this is chromosome number 2, and that's where these two variants and this gene lies on. These two exact same variants are also splicing QTL variants, regulating a gene called PTPRC on chromosome number 1. Okay, so different chromosome. So you see on the figure on the right, whenever we have a cis eQTL for this gene, we have a trans sQTL for PTPRC. Okay, across all these cell types. So then what happens is that this gene, which is the cis E gene, is called HNRMPLL. That's a classic splicing regulator. Okay, that's an RNA binding protein. So it produces this protein, and this protein will come in and bind to exon 4, 5, and 6 of this gene called PDPRC, and then produce two different isoforms, the RO isoform and the RE isoform. So because we have single cell data, we can actually stratify the cells based on the expression level of HNRMPLL. If they don't express HNRMPLL, then the PDPRC is not spliced. And if they express HNRMPLL, then PDPRC is more spliced. Okay. So this is really interesting to us. But then we asked one further question. For those of you who study immunology, you would know that PDPRC also has a different name, which is called CD45. Okay, that's a classic surface marker for T cells. And then these two isoforms are all an RA plus isoform. They decide whether the T cell gets activated. So then we ask one further question. Do these two variants actually change the T cell proportions? And the answer is yes. Right? So these two variants also changes the naive to activated T cell proportions. And the p-value is highly significant. So just to sort of to wrap up this part of the story up, we identified a mechanism which is independent regulation of the same gene in two different cell types, which produces a protein that regulates in trans the splicing of CD45, which is a classic surface marker for CD4 T cells, which then regulates the CD4 T cell proportions. And this is only using single cell data, right? We don't have any protein data. So I think the single cell data in population scale is extremely powerful. The second story I'm going to tell you is related to diseases. Even though the people that we sampled is apparently healthy, we can still use their genetic information to infer disease-associated target genes. So what we did is that we performed colocalization and Mendelian randomization against these 5 different diseases. AD is, in this case, AD is atopic dermatitis, asthma, Graves' disease, rheumatoid arthritis, and lupus. So for the sake, okay, I'm going to describe you the figure. The x-axis are different cell types. Y-axis is different genes, and then the color represents different diseases. So we identified somewhere between 30 to 60 targets, but for the sake of time, I'll just describe you one of them, which is this gene called TCHP. So what we found is that this gene, whenever there's a splicing QTL, it would colocalize with GWAS. Okay, it would colocalize with GWAS for Graves' disease. So this is GWAS for Graves' disease. And then the CD14, there's splicing QTL in CD14 monocytes, CD16 monocytes, natural killer cells, T cells. And we will see when we go to B cells, the signal disappeared. Okay, so you see on the left side, out of the 22 cell types, only 1/3 had a splicing QTL signal, and 2/3 of the cell types don't have a splicing QTL signal. That means that if we have looked at this in a bulk tissue sense, we won't be able to recover this because it will be masked by the cells that didn't have the signal. So then we went and validated this using a mini-gene experiment. This is nicely contributed by our collaborator, Donald Sim and Xavier Roca. So what we did is that we cloned out this exon. Actually, before I tell that story, there's something nice here that I want to tell you. We got really lucky for 2 reasons. This variant is sitting on a splice site, right? So it's we have confidence a priori, and also this variant is an Asian-specific variant. So you see that this variant has high allele frequency in East Asian populations, Japanese, Chinese, and Korean. Lower allele frequency in Southeast Asian, and then it disappeared in East Asian populations. Okay, in South Asian populations, excuse me. And we can replicate this effect using 1,000 genomes. So now let's come to validation. We cloned out this exon number 4 and its surrounding introns using a mini-gene construct. So we did 2 mini-gene constructs, differing only at the base, at the RS744. So we found is that if we have a G allele, which is a protective allele, we get normal splicing. If we have the A allele, it destroys the splice site and then leaves the intron retention, which removes the transcript because of nonsense-mediated decay. Okay. So this paper was published as a cover article in Nature Genetics, but this paper had a few problems. Okay, which we only realized after publishing the paper. So first of all, this method, we're still using pseudobulk, right? And so the natural question is, pseudobulk would actually lose power to call QTLs. And also because it takes the average of many repeated measurements. And also there is, these pseudobulk methods is based on previous methods established for bulk studies, which does not account for single cell specific artifacts. Okay, so there's 5' and 3' bias. There's high PCR duplication. And just take a look. Excuse me. So, in fact, we surveyed all the existing literature. There's a number of papers doing single-cell eQTLs, as I mentioned before. And there's a number of papers doing bulk splicing QTLs, right? But a single-cell splicing QTL method is actually still missing. So the second reason, which I think is actually more important, is that when we do pseudo-bulk single-cell splicing QTL work, the cell state information is actually masked when we do pseudo-bulk. And this is important because people like Soumya Richarduri at Harvard Medical School have actually shown that this is actually a quite important regulatory effect. What they have found is that within T cells, for example, when you have different states of the same type of T cells, they would have different genetic regulatory information, right? So then we developed this method called ISSAC, which is called integrative single cell splicing analysis and sQTL caller. And it addresses exactly these two problems. The first one is how do we map cell state dependent sQTLs? And the way we do this is that we don't do pseudo-bulk anymore. We started by trying doing single cell, but the matrix is too sparse and it's just too noisy. So then we decided on doing metacell, right? So we merge the k-nearest neighbors into a metacell and use that as our unit of measurement. And now, so for each individual, we don't have only one measurement. We have multiple measurements equal to the number of metacells. Now we're faced with a different problem, which is that we have a two-layer statistical model, right? So the first layer is the number of is the metacells, and the second level is individuals. So then we account for this using a genetic relationship matrix, right? So it's a block diagonal structure. What's really nice about this is twofold. A, we reduce the false positive rate. If we don't control for these relatedness, we will have severely inflated p-values. The second is actually quite nice, and it's a byproduct, is that we can account for genetically related individuals, such as siblings or parents and children. Previously, we have to throw all these individuals out. But now, because we have this genetic relationship matrix, we can keep all these individuals and control the type 1 error very, very well. And the second is how do we mitigate single cells RNA-seq artifacts, especially 5' and 3' bias. So you'll see that when you're doing 3' or 5' single cell RNA sequencing, you'll typically have this kind of restructure, right? A read distribution, where most of the reads are either distributed around the TSS or around the TES. So so the way we do this is we first do cell barcode UMI-based read collapsing. This means that we reduce the number of duplications in PCR because single cell has a lot more PCR cycles than bulk. So we reduce and collapse the reads. And also what we do is that instead of doing junction-based quantification, we do splice site-based quantification. And the reason why I'll show you one slide later. But this slide, I'll show you the definition of SSUR, which is splice site usage ratio. So let's say we're interested in the splice site C. So I don't know if you can see, but here is A, B, C, D, E, and F. Okay, so suppose that we're interested in splice site C. The first thing we do is that we quantify the junctions that include splice site C, which is I1 and I2. And then we quantify the junctions that could have used splice site C but excluded it. In this case, for example, splice site E and splice site F could have paired with splice site C, But then this splice site F instead chose to skip splice site A. And also, there's also other splice sites that are just not relevant, right? And we denote them as N. So then the SSUR is defined by the reads that includes splice site C versus all the reads that could have used splice site C, okay? Include those ones that excluded splice site C. So what is the advantage of doing this instead of doing junction-based reads? So this, I hope this slide will make this clear. So let's say, so what I did was simply blow up the read distribution, yeah? So most of the reads you see will be distributed around the 5' end, okay? So let's say if we use leafcutter, which is the most popular bulk tool, splice QTL tool, what they do is that they would quantify I1, and as the denominator, you will have I1 plus I2 plus E plus N1 plus N2. So basically all the read in this cluster. However, what you see is that because N1 is towards the 5' end, you will have more reads. This would actually dominate your denominators. Eventually, what that does is that your splice junction read, IUR, will no longer be linearly correlated with actual transcripts because of this 5' bias. You see that on the x-axis, this is the ground truth. This is our simulated data. On the x-axis is the ground truth. On the y-axis is the splice site, is the IUR, intron usage ratio recovered by leafcutter. You see that there's almost no correlation between the ground truth and the recovered IUR. Alternatively, when we use splice site usage ratio, because we throw out all these non-relevant splice sites, now our recovery is almost linear with the ground truth. So how about on simulated data? Our node simulation, our calibration is great. So is leafcutter. So kudos to them. But when we compare power, because leafcutter leaves out so many information on the table, their power is not great, especially for 5' or 3' bias single cell data. So we can gain as much as, which I'll show you on the next slide, which is as much as 150% gain over the of power. So this slide shows on real data, right? So we use 3 different types of cells: abundant cells, medium cells, and rare cells. So in abundant cells, we can increase the power by 40 to 60%. In medium cells, we can increase the power by more than 100%. But the real advantage lies in the rare cell types, right? Which you start off not having enough power. And that's really what you want to get power from, increase from. So here we get somewhere between 130 and 150 power increase over bulk, pseudo-bulk sQTL methods. But one remaining question is this. The reason why people like pseudo-bulk is because they're fast, right? They're linear models and they're extremely fast. And if we do this using, let's say, commonplace methods called, for example, Glimmer, which is one of the most popular R package, it will be extremely slow. It will be 100 times slower than the linear methods. So what we did was 2 things. The first thing is that we wrote our method entirely in C++, which is already 10 times faster than R. And the second thing is that we used the past 50 years of statistical innovation. We didn't invent anything, right? It's all common knowledge in a statistical field to make our method extremely fast. Okay? Because, for example, we can do PCG, and we don't have to invert the matrix, which saves us a lot of time. So eventually, our method is about 10 to 100 times faster than LME4. Okay, so basically, our method is the same speed as if you're doing linear models. So 5,000 individuals with 1 million cells, 5 minutes per splice site using 1 CPU thread, but you can spread it over 100 CPUs, for example. And if this is not fast enough, we have a GPU version, which is coming soon, which is, again, 10 times faster than a CPU version. Okay. Okay, so then we can apply this method to Alzheimer's disease. So we apply to ISSAC to dorsolateral prefrontal cortex single-cell RNA-seq data. So we apply this to about 3 million cells from 722 specimens. So we classified them into 7 different cell types, including excitatory neurons, inhibitory neurons, oligodendrocytes, progenitors, endothelial cells, microglia, and astrocytes. We identified 29,000 sQTLs for protein coding genes, and then 4,000 for link RNAs. And most of these splicing QTLs are independent from eQTLs, right? So there's about 30% overlap between eQTLs and sQTLs, but even within those overlaps, most of them don't share the same causal variant. 75% of them don't share the same causal variant. Only 25% of them do. So the question is, what are those 25% that actually share the same causal variant? So what we found is that intron retention plays a big role, right? So if you have a variant that disrupts a splice site, and that causes intron retention, then that usually leads to frame shift, and then that leads to nonsense-mediated decay. But one nice thing, so I guess going back to comparing with leafcutter does not quantify intron retention for a very simple reason. Because intron retention, there's no intron cluster. There's only a single intron. A leafcutter needs to have a denominator, which means that it has at least 2 introns. So leafcutter wasn't able to quantify intron retention. But using splice site level, we can actually quantify intron retention, which is a huge advantage when we're talking about comparing eQTLs and sQTLs. So basically here, SSUR becomes the reads that included the splice site divided by all the reads, right? So then we were able to identify several interesting intron retention signals, including this gene called NAGK. So for this gene, there is a variant which is in the exons, changes from C to a T, and then this changes this causes intron 9 to be retained, which causes premature termination codon, which leads to NMD. So that's why you see, there is a excitatory neuron splice QTL, which has very significant p-value, which then leads to a eQTL, which is of less significant p-value, right? So this is an intron retention effect. Okay, so now the second question that I talked about is cell state dependent sQTLs. So we focused on one cell type, which is inhibitory neurons. The reason why we do that is because inhibitory neurons itself is very diverse, and also there's sufficient number of cells, about 500,000. So we can infer cell states in many ways, but here we're just using the most simple way, which is the principal components. So this is PC1, PC2, PC3, PC4, PC5, PC6, PC7, PC8. For the sake of time, I'm just going to show you PC1. So for PC1, this principal component is related to neurogenesis and also generation of neurons. Of course, they're just not synonymous. And what we found is that in different states of inhibitory neurons, there are genes that has differential genetic regulation, right? So for example, for this gene, PREX2, in this lower PC loading state, the variant G decreases the splicing level of a certain exon, whereas in medium and high PC1 loading, the same variant would actually increase the inclusion level of this exon. So we found in total 370 cis sQTLs to be cell state dependent. And also we mapped genotype by disease interactions. And so there are some genes that are only showing up in Alzheimer's disease. So here AD means Alzheimer's disease. And what's interesting is that this MT1X gene is also cell type specific, and it's cell type specific in astrocytes, which is one of the rare cell types, or least abundant, less abundant cell type. So we identified a total of 480 biased sQTLs in around 200 S genes. So again, going back to disease, for the sake of time, I'm going to, so this is the same figure, same figure style that I showed last time. So I'll just skip explaining the axes, but I'll show you one example, which is the TRPT1 gene. So you see that this gene is co-localizing with neuroticism GWAS across all these cell types. And then this variant is actually, we're less fortunate with this variant because it's not sitting on a splice junction. It's actually an exonic variant. But still, we apply the same technique. We cloned out, this time, exon 4, exon 6, 7, and 8 of TRPT1, constructed two mini genes. This is done by Zhiyang, my postdoc and lab manager. And with these two variants, the G variant would give you exon inclusion, and the A variant would give you exon exclusion. Okay. So that paper is also, we think that study is also very nice, but it's still facing some problems. Okay. So we're going one step deeper. And so what is the problem with that paper? Single cell is still missing a key information, which is cellular microenvironment, right? This is especially for solid tumor like the brain. And also cellular microenvironment it can only be captured by spatial assays, but they're too expensive and they do not scale to large sample sizes. So then our hypothesis is, could we actually harmonize published data across labs, right? Because great many labs are sequencing dorsolateral prefrontal cortex, right? They're just not talking to each other. They're depositing their data in public repositories using different technologies, but they're not talking to each other, not collaborating with each other. But can we serve as the middle layer and integrate all that data together? So there's a fundamental problem in data integration, right? And there's actually two fundamental challenges. The first one is called vertical integration, and the second one is called horizontal integration. I'll show them both. Just take a look. So for horizontal integration, so actually let me explain this figure a bit. On the x-axis is the different cells. On the y-axis is the genomic features, right? So if we're doing horizontal integration, then we're integrating across different batches within the same modality, right? So in this case, we're integrating batch 1, 2, and 3 for their RNA expression levels. So this is basically equal to removing batch effects and aligning cell type annotations across data sets. They use genomic features as anchors. One example will be cell type transfer from a reference data set, which is annotated, to a query data set, which is unannotated. So there's a number of methods that's doing this. In the single cell field, there's ST-MAP, Harmony, Scanorama, Surat, and ScanVI. And I think now there's more. And in spatial, there's a method called Stellar. You can also do it in a horizontal way. In a vertical way. In a vertical way, you would align the same batch of data, but across different modalities. There's basically we'll be removing batch effects and align multiple omics modalities using cell as anchors. So one example would be joint analysis of spatial ATAC and RNA-seq data. And in this space, there's also a number of methods. For example, Liger, INMF, glue, multi-VI. And in spatial, there's spatial glue and meso. So if you look at these two axes in this figure, there's methods doing vertical and horizontal integration. But for bidirectional integration, there's one method for single cell, which is Surat version 5. But at the time we were writing this paper, there's no method that does bidirectional integration for spatial. For us, so we asked a very naive question, is that have people solved this problem before? And it turns out that people have solved this problem repeatedly in computer science. which is a classical domain adaptation problem, right? So horizontal integration is like integrating photos with different white balances. And then vertical integration is pictures that's different styles, right? But they're basically measuring the same object with different styles or different backgrounds. So, and both of these are special instantiations of a more general problem, which is called domain adaptation. And this problem has been solved repeatedly in computer science. For effective domain transfer to be achieved, predictions must be made based on features that cannot discriminate between the reference and the query domain. And this paper has been cited almost 10,000 times. So what we do then, so this gives us clarity on what to do, right? So our method is extremely simple. The idea is extremely simple. We first use a graph neural network to extract the latent information. And then we just do prediction like all the other methods, right? So for vertical integration, we predict the reconstruction using a VAE, for example. And then horizontal integration, we predict the cell type labels, right? But we have an additional head, which is to predict the domains. And the domains are well labeled, right? For batches, we use the batch as the domain labels, which we already know. And for the modalities, we use the modalities as the domain label, which we already know. And using these labels, we can design an adversarial trainer, which feeds back to the latent representation and train the latent representation to forget about these labels, right? So this basically forces the latent representation to only focus on the features that are descriptive of the cell types, but forget about the information about the batches. So let's see how this method compares against other even more complex methods. So we start with this manually labeled embryonic day 10 mouse data, which is to integrate RNA and protein. So that's the ground truth. This is using DIRAC. This is using multi-VI, which uses embedding. This is using MaxFuse, which is using embedding and alignment. This is using spatial glue, which uses embedding and spatial. And this is using our method, which is called DIRAC, right? Domain invariant representation through adaptive calibration. So you see that using through this gradient, our proof gradually increases, right? As we add more information to our model, our model performance improves. And you see that a lot of it is because we can integrate data better than existing methods. Right? So for the sake of time, I'm going to fly through some of these slides. So for horizontal integration benchmarks, I'm sure you've seen this multiple times. This is one of the classic benchmark data sets. So if we use SVM and SCMap to annotate this slide, there will be missing layers. There'll be missing layer 2 and 4. If we use Stellar, AdaBoost, and ScanVI, they will have all layers, but the layers are fuzzy. But if we use DIRAC, the layer is sharp, and it corresponds very well to the ground truth. So we can also do this in intestinal data. So this is the ground truth manually labeled, and this is the DIRAC prediction, which is almost indistinguishable from the ground truth. So then we generated a novel data set using a spatial multi-omics data method, which is stereosite-seq, which has RNA and protein, which is subcellular resolution. So we're able to use DIRAC to map out the entire spatial migration process just using one slide. So here we took the mouse thymus and stained them with DAPI stain and also used stereosite-seq to get the RNA and proteins. So here, using the vertical integration capability first, we were able to map out the spatial domains, right? So we map out the capsule, cortex, CMJ, which is the cortex medulla junction, and medulla regions. And we can also do a final resolution, but for the sake of time, I'm just going to skip the right side. And then using the vertical integration capability of DIRAC, we can transfer the label of a single cell site-seq data to the spatial site-seq data. And for the single cell site-seq data, we can actually also get the pseudotime, right? So we know that, on the single cell scale, if we start the root as double negative cells, it goes into double positive cells. and then double positive quiescent, double positive signaling, and then turn into single positive immature and single positive mature. Single positive just means CD4 and CD8. So, but we can layer that on top of our spatial data because we can use that single cell data to annotate our spatial. So now we get this beautiful spatial map, which is we start with the double negatives close to the capsule, and then we generate, they will turn into double positives, and then they will go through 2 cycles of proliferation, right? And because we have single cell resolution spatial data, we can actually count the number of cells, unlike the deconvolution methods. And eventually they will become single positive and they will move towards the CMJ. And eventually they will become the single positive mature cells, CD4 T cells and CD8 T cells and DIRACs, which occupies the center of medulla. So just to sort of summarize, we've had, these are some publications from our lab in the last few years. So today I talked about 3 papers, which is the Tian et al., the Xu et al., and then the ISSAC paper. I didn't have time to talk about the other papers, but I'm happy to discuss them in privately or after the talk if you're interested. So this work are supported by our grantors and also our generous collaborators who share the data with us. And we're always hiring PhD students and postdocs. And if you're interested, feel free to take a picture and feel free to reach me through email. Thank you very much.